Hello, my name is Cristina MacSwiney, Maria Cristina MacSwiney Gonzalez. I'm a researcher in Mexico, and today I would like to talk about this project, the Acoustic Library of Mexican Insectivorous Bats, the project Sonozotz, AMMAC, CONABIO, which is a national project that we carried out a few years ago, but that continues right now with other students and other researchers still working with echolocation in Mexico. We already know that the bat species forage in different sites, in different locations, and the place where they forage is related to the wing and echolocation calls they emit. For example, the bats that forage in places outside the vegetation, in the open, like molluscs, for example, they tend to have like narrow and long elongated wings that are not able to maneuver between the vegetation. And the collocation calls of these species, normally there are low frequency, long duration, and very intense collocation calls. In contrast, we have other species that normally forage in between the vegetation, in between the trees and branches and so on, and a lot of obstacles. And they normally have FM echolocation calls. And also the wings are more narrow and short. So the design of the wing morphology and the collocation call is extremely related to the place where these species forage and find the food that they take every single night. When we analyze the collocation calls to try to assign an identity of this echolocation to a certain species, we normally measure echolocation calls in different ways. One of the most important characteristics is the peak frequency, which normally is related to the sensitivity that this bat has in the ear to the collocation calls. So the frequency, the peak frequency, help us to identify with some certainty the species that produces these echolocation calls. This is, let's say, the most important measurement. But we also have different other measurements that are important, like the call duration, the start frequency of the call, the end frequency of the call, the distance between one pulse and the other, and so on. So we normally use these measurements to try to assign the identity of the bat emitting these echolocation calls. But we have been working with bats and recording these bats and trying to identify the species, most of the time releasing the bat from our hands, or releasing the bat in a fly cage, for example, and recording the bat so we can know how this bat produces their own call, and in the future to use these calls as references when we have free-flying bats and try to assign this unknown identity of a bat when we record free-flying bats to a certain species. And for many years, this is a recent map showing a little bit all the countries in Latin America that has efforts to produce acoustic libraries, but this is a recent map produced few months ago. Many years ago, when we started the project, this map didn't look like this, and although we have this map with in blue several countries in Latin America, what we really know is that most of the libraries are regional efforts or very localized efforts. We don't have, in general, in Latin America, national efforts to organize or to produce an acoustic echolocation bat library of bats as a national effort itself. In Mexico, let's say this is the first proper effort to produce a library. This was published in 2002 by Raidel Arita, Dr. Hector Arita, and other colleagues in Mexico. And this project was carried out in the Yucatan Peninsula, which was extremely helpful for me when I was doing my PhD, because I was trying to assign the diversity of echolocation calls in the Yucatan Peninsula. And this paper was really, really useful for me. However, this paper does not include the most of the diversity of bats. So I have to record my own bats in 2002 and 3 and 4 when I was working in the Yucatan, and produce my own library. But this paper for sure was really, really helpful. And this, from my perspective, is the first echolocation call library paper produced in Mexico itself. So for many years, the researchers that attend to the National Mammalogy Meeting, which is carried out by the Asociación Mexicana de Mastozoología, AMMAC, we were talking about this need to produce an echolocation bat library in Mexico. And we couldn't, we talk about this in the international meeting and so on, but it's just in 2012 when we had a bat symposium that many researchers working with bats talk about seriously about doing this. This is the initial approach. And then in the following conference in 2014, we had a group of researchers already organized and trying to divide the country in different regions to be able to do this project. Because as you can imagine, Mexico is such a large country, so diverse, that we need to have a protocol, that we need to get a lot of people involved in this project if we want it to work. So in the meantime, between all these meetings, a lot of us were producing results of the echolocation calls, but no matter what, we could see that we need a national effort and to be organized to be able to carry out these, or to construct this library that we were wishing to have. Because why we wanted this? Because we could see that there was potential variation of the echolocation calls all over Mexico, that some calls that were recorded in the state of Oaxaca look different to the ones we got in the Yucatan, or because many of the species that we have in these regional libraries were not available in other parts of Mexico, and many species in Mexico were not characterized in terms of echolocation. So there were many reasons for us to do this. Particularly important if we consider that, for example, in Mexico, all these wind mills and wind parks that are approved need an echolocation study, and how these people that are managing the resources are able to produce these reports if they don't have a library to match whatever they're recording. So that's an example. So this is the name of the project. And I have to say that at this time, in 2015, more or less, Dr. Miguel Briones, which was the president of the Mammal Society, approached to the commission, the National Commission for the Knowledge and Use of Biodiversity, CONABIO, which was a governmental office dealing with diversity, and they were already carrying out a project recording diversity, not only bats, but birds, insects, and environmental noise as well, in several habitats in Mexico. So they were gathering all this information, but they didn't have any library to match at least the echolocation calls of bats. So that's why they approved our project. So what we did, or the objectives of the project, was to create this library, having groups organized all over Mexico in terms of habitats and diversity, to have these workshops, to first organize a protocol and then to replicate it in another regional workshop, so everybody learn about this protocol. one of the goals as well was to try to organize all these pre-existing calls from different colleagues in Mexico. And of course, to elaborate a common protocol to record, release the bats, and obtain these echolocation calls, to validate this call and storage them in a server from CONABIO. And also something that is a work that is still going on, which is to analyze the calls and discriminate species and at national or regional level, something that we're doing right now. So what we did is to divide the country, which is a large country, in 8 regions. What you can see in white over there in the map, some states that were not sampled, and not only for safety reasons, but Mexico City, that is in the middle of the country, could not be sampled, or was not sampled, because we have an earthquake, particularly during the days that we were going to do the sampling over there. So the sampling in Mexico City was canceled because the consequences due to the earthquake in 2017. But the other states were not sampled because it wasn't safe, I have to say. This was the group that got together in Mexico City once the project was approved by CONABIO. It was the board of the Mammal Society, along with the people from CONABIO, and all these 8 people in charge of every region in the country. And in this important meeting, we decided that we were gonna do a protocol and a workshop in a diverse place. And we choose La Mancha, Veracruz, which is in the Gulf of Mexico. And it's an important site because it has a diversity of bats, insectivorous bats that were catch outside roosting places, but also trolling bats that were flying in the open. So we can try the protocol and to see if it was working properly. Because after that, we moved to different regions and we train people all over Mexico to be collaborators of this project. What we decided to do is to record the bats, but also to have ID of the call, but also voice notes, to relate the environmental situation, every situation when we were recording a bat. How was the weather? How was the technique to release it? It was hand release or recorded in a fly cage. Everything is written down, but also it was explained in a voice note. And also we did a monitoring, which I will explain later. And we got photographs of each individual along with tissues, which are important because that allow us to verify the identity of some species, which is really important because we have a lot of meiotis, we have a lot of species that are not so easy to identify in the field. As I mentioned, we also got an acoustic monitoring, 15 minutes each night. Not all the participants did this, but I did. And that was really important, for example, for a thesis of one of the students working in the Yucatan Peninsula, a thesis that is already finished and about to be published. And we were also taking photos of the wing shape of the bats, as you can see in the photograph. And these wing photographs were really important, at least for me. We published in collaboration with Sandro Spina Garces, a researcher that works in morphometry, a project relating the wing morphology and the foraging gills of bats in Mexico, which was already published 2023 last year. And we also got the environmental variables, which are also important to relate the echolocation calls, because we know that the echolocation calls are affected by humidity and temperature. So many of the works of the students and other research that we are right now doing is related to the environmental variables that we did record every single night over there. This is just an image of many of the places I have the privilege to go. We were sampling in the streams, in cenotes or petenes, like the freshwater sinkholes that are in the Yucatan Peninsula, in thatched roof, in Maya ruins, caves, urban sites. And I have the privilege, I have to say, to go to the Sea of Cortez in the north of Mexico to sample this particular bat, a fishing bat that lives in islands in the Sea of Cortez. So I was really lucky to be able to be there. This is a paper as a result of this national effort. It's published in 2020 in Ecology and Evolution. You can download it for free. And in this paper, basically, we report all the results of the project. How many bats did we catch? The location of the sampling sites, how was the taxonomical coverage, how was the distribution coverage of each species. So it is basically the paper with the results of the project. One of the most important things that we have to deal with is how to storage all this information. And for this, I have to say that we were really grateful with the people of CONABIO. CONABIO, this governmental agency, helped us to, they gave us a server, but also all the platform, they create this platform where we can download or upload, every single bit of the information we got from bats. The identity, the sex, the measurements, all the collocation code, the voice notes. We could see the sonogram of the bat with the photographs, all the sheets from the field when we take in the notes, everything was uploaded in this platform. And the good thing was storage in a server from CONABIO, but also storage in every computer of the regional manager of the regional responsible of the echolocation library. And we could as soon as we upload the information, we could share it with the server in Mexico, so we don't lose any information we upload. This is really key, and this is something that is really difficult in terms of storage because echolocation libraries or echolocation recordings are really heavy. So this server from CONABIO helped us a lot in terms of storage. At the end of the project, we got this amount of recordings from most of the Mexican insectivorous species. We have so many sites, 183 sampling sites in 27 of the 32 states, many tissues that are available for whoever wants to with genetics, an ID of the species that we collected. So these tissues are available in a tissue collection. And we also have photographs that are shared in the CONABIO web pages like Enciclovida and iNaturalist. This is some of the results. We have a book. Everything here is available for free. We have a video in YouTube, in the YouTube channel of the AMMAC Society. We have this book, which is in English and Spanish, and also this manual that is sharing the information how to construct an echolocation by library, which is also in Spanish and English. So I really invite you to download these products. We're continuing with the results. I personally recorded several phylostomids, and we're analyzing these. We could see their phylostomids, no matter if they are really similar in the collocation codes, they show some differences, and we're publishing this really soon about the results of the many frugivores, nectarivorous bats that we recorded in this project, and this is about to be published soon. And one important, and I will finish this talk talking about Latin America. Latin America lacks many studies in acoustics and libraries as well, as we have seen in the previous chart when we were talking about acoustic libraries. And many of the studies are really, really specific. So what we really like to do in the following months and years is to gather all these echolocation codes of insectivorous bats that are missing in Latin America that we haven't recorded yet, and share them all together. And for this, we're getting these examples from other libraries like this in Brazil, but we're getting together a library that is with the help of Wildlife Acoustics that is providing many equipments particularly for several countries that they don't have the money to buy these devices. So Wildlife Acoustics supports this project. This project is ongoing, just started in the COLAM meeting in the Latin American Bat Conferences that was 3 weeks in Cusco. And this is the logo of the conference. And we're right now getting all these researchers involved in different countries in Latin America. I was one of the teachers of the workshop, and we are sharing, of course, our experience and getting all the Latin American colleagues to do this project. So in the following months and years, we're going to have this effort, Latin American effort, to have a library. I just want to thank all the people involved in the Mexican echolocation library, because as you can imagine, it was so many people, hundreds of people, students, people in the field helping us to get to the caves and Maya ruins and so on. Obviously, my students, colleagues, family, because otherwise you cannot do this research without the help of all the people around you. So I would really like to thank Wildlife Acoustics to invite me to this chat, to this talk. And I'm more than open to get any answers from your questions. Thank you.
Wow, that was brilliant. I can't believe how thorough that acoustic library, well, that library is. I mean, it's just immense, the information that you've got there. That is fantastic. Thank you so much for your presentation as well. You're getting lots of claps and hearts and thumbs up and everything else. You inspired loads of people, I think, from all over the world. And we've got questions. Questions are coming in. So I'll get straight to those. How many Mexican or Latin American bats are there that have not had their calls recorded or characterized yet? And are there any species where this is urgently needed for their conservation and monitoring?
Well, as we report in the paper, most of the molluscids were missing. Not all of them, but I don't know. In Mexico, we're missing like 30% of the species, the insectivorous species, because they're really hard to get or catch. Basically, molluscids are the most obvious group because they tend to roost very high. A lot of them roost in cities, but on the top of churches or buildings that are really difficult to get. And also, we got some of the species of molluscids, but they're really difficult to record because they need to be in the open, they need to be stress-free to produce the best echolocation calls. And for some species, we did catch them, but we caught 1 or 2 individuals, and they were not producing the best echolocation calls. So even some of them were caught in the project, we didn't obtain good calls. So for that group, we need to focus our efforts and I don't know, try to find the roosting places, try to catch as many individuals as possible, not having just one lucky individual in a misner in the middle of a pond, but to go to the roosting places. I think basically that's the answer. And of course, all the philostomids that were not included in this project because CONABIO were funding this project for insectivorous bats, but we did catch many frugivores in Mexico, and nectarivorous species, sangivorous, whatever, and we did record some of them in many regions. So we're gathering all this information for a further publication really soon.
Now, the one thing that inspired was your last words. Are you thanking people, and you thanked your family and your friends, which I'm assuming that you drag into some of this research that you. So, but obviously you've got these research teams that are out there that are collecting data. Are they using citizen scientists as well? Are they just using people that are just interested or just roping in their family and friends as well?
Right now we're not doing any citizen science to record bats like in this project, but we do get a lot of information from citizen science. For example, we have this platform that is also run by CONABIO, which is a naturalist. In Mexico, it's called naturalista. And that has been really helpful to find, for example, bats that are not easy to see. I remember like a couple of years ago, somebody from the state of Hidalgo shared an image of a Euderma maculatum. This is a bat species that is really, really difficult to catch in Mexico. And it was seen in a frame of a door in a house in the state. So a lot of Mexican researchers ran to this city to try to find this specimen, this individual. So examples like this, we're using citizen like science from the citizen itself, just to try to see some new location, new caves, roosting places that we didn't know, and we need to maybe go to these places, catch the bats so we can obtain the echolocation call from these bats. So that's a way it's helping us. Right now, it's not helping us recording bats, but the idea of the project as well was to do this national monitoring in the future. And for that, we have seen the sample from UK that is really good, working perfectly for several years. So we do want to implement this in Mexico as well.
Well, it's funny, just going on from what you're talking, there's a couple of, they're not really questions, well, they sort of are questions, but you've inspired people, basically, is what's happened here. Fantastic presentation on Mexican bats. We're based in Yucatan and have access to recorders and funds to deploy the recorders and recover the data. Who shall we talk to? And then somebody else, how can someone get involved with these endeavors in South America? So in other words, how do people get involved? You want more people, they're here somewhere in this room. How do they get involved?
Well, the project right now, the project Sonozotz, let's say it's over because we're not gathering as project together all this info. However, all the people that I was mentioning in the regional Mexico, in the regions that we divide the country, is still gathering some calls. The idea will be, I didn't mention this, but I have to say it right now. One of the problems we have as a project was at the end of the project, what to do with these echolocation calls, where to upload these to be free, because this was paid by the government. So it has to be free for any citizens that want to download the echolocation calls. However, just in the moment when we finished the project and we were about to release the platform, we had a change in the management of the CONABIO office. And that new administrative team didn't put any effort on this project. So we're still talking with the people involved in CONABIO with us, related to us, or collaborating to us, because we have already the platform. And we were just in the moment to make it look nice, let's say, with a web manager, to make it look nice and fancy and attractive. We were there because the machine, let's say, where the skeleton behind was already done. It was just the imaging what we were working on when they have this difficulty. But you were mentioning before Xenocanto, or I put a slide of KetoBox, and I'm really in touch with CONABIO saying, okay, if we're not having the chance to publish this as a country, as this governmental effort, let's do it somewhere else, because a lot of people contact me, from time to time, and I feel embarrassed not to given the information, because I have to have the approval by the government. No matter what, I did the recordings. Normally, when I recorded the bats, I shared with them. But if it's somebody else's data, I cannot. So this is a problem we have right now, and we need this is a problem we have to solve, because it is a pity that we have more than 2,000 calls and people recording right now and storaging more and more and more echolocation calls, particularly for this species that we only have 2 or 3 calls, and we need to share them.
Yeah, and the thing is, it seems like a blockage for you, because you're only going to get more data. I mean, that's the problem, isn't it? We can collect more data, and the data is easier to collect. And the more people that we have. So yes, I really understand, and I feel for you, and hopefully it'll be sorted soon. Fingers crossed. Anyway,
Yes.
Loads more questions on a complete range of things. Do your acoustic devices also record temperature, humidity, other environmental factors? Or do you monitor that separately? Do you monitor those sort of things?
Separately, yeah, we did separately.
Um, that was an easy one.
Yes.
Can you recommend how to accurately record sounds of a single bat from a colony of bats in their natural habitat? What is the directionality of the microphone to avoid picking unwanted sounds? Here's a tough one. Yeah.
That's a very tough one, and it's barely impossible in the tropics, because you have normally so many individuals coming out from a cave or a shelter, you know. So the ideal situation will be to catch them and wait to the emergence to be kind of over, but it's never over in the tropics. You always have other bats flying around. So you have to be very accurate when you release, if you hand release the bat, when you release, and to be really ready with a notebook to be able to write down at what exactly time did you record the bat, because otherwise you will have always individuals from other species or maybe the same species flying around, and it can be really confusing if you're not familiar with the echolocation calls and the other species that are flying around. So the idea will be to hand release the bat in an environment when it's not so crowded, let's say, of noises. Maybe a little bit late in the night, but you have to be sure that the bat is not stressed and not, after being held for many hours. So the idea would be to release the bat as soon as possible, but to avoid this emergence time when you get a lot of calls.
Yeah. Yeah. We tend not to have that problem so much.
Yes.
uh, uh, so this is somebody from India who's interested in building an echolocation library for my study site in India. Is it enough to use only acoustic tools to gather data to gather the diversity data of all the echolocating bats in the area? I'll be surveying different habitats. Is there other things I have to consider?
Yes, in the project, well, this is an echolocation call project, but of course, you need to go to the roosting sites to sample with mist nets, because in these tropical countries that we have species that feed on flowers, and you have to get all the diversity through different methods. And of course, roosting sites is really important, because some bats that feed on flowers and fruits and whatever, they're not so easy to catch because they're roosting in vegetation. So you need to get bats from that roost in caves and houses and bridges and different scenarios, but also you have to get the diversity from other species that are not recorded easily through echolocation. For example, in India, you will get theropodid species that they're not echolocating, but they're part of the diversity, but they have to be caught in the vegetation where they're foraging with fruits and flowers and so on. So you have to get different methods to get the diversity in the tropical areas.
Yeah, like your knowledge of this is just astounding, and I think but we've run out of time. There are still some more questions in the Q&A if you would
Yeah, I
have.
will go through them.
Yeah. And I think there's been a request in the chat. I've been trying to follow lots of things for the papers that you mentioned as well in the in your presentation. If they can be accessible in some way, that would be wonderful. Just the location, just so people can go and access them. But
Yeah,
I want to thank you so much, Cristina. That was amazing, and you've inspired loads more people, which is what we like in the bat world. Thank you so much.
thank you very much.